am going to teach you Eubacteria. So what are Eubacteria? Actually, Eubacteria are the true bacteria which are having rigid cell wall. Some are motile, some are non-motile. Some Eubacteria are motile. They are having pipe-like structure called flagella. If single flagella is there, then we call it as a flagellum. Because flagellum is singular, flagella is a plural term, and we know that flagella which provide motility or help in movement. So these are true bacteria. Cell wall is present. Now, under Eubacteria, first we are going to study about blue-green algae or cyanobacteria. Blue-green algae. Now, their habitat, where these are present. These are present in wide variety of habitats such as polluted water, sea water, and even hot springs. Now, they are predominantly found aquatic, but some are terrestrial also. Now, we are going to discuss about the structure of cyanobacteria. If we talk about the structure of cyanobacteria, they are having cell wall, it is a cell wall. It is also double layer. Inner layer is made up of peptidoglycan, and inner to it plasma membrane is there. And this is made up of lipid and proteins. They are having gas vacuoles, but remember, membrane bound, although it is a bacteria only, membrane bound cell organelles are completely absent in cyanobacteria. They are having different pigments. That is why they are photosynthetic in nature. Different pigments are there, and these pigments are present in thylakoids. These are membrane bound organelles. We can call it as a lamellae also. Here actually these pigments are present. This type of structure. And here the circular chromosomes or circular DNA, that we can call it as a nucleoid, is there. So, and now what are the pigments present in it? So we can say chlorophyll A, then beta carotene, xanthophyll, C, phyco. C means for cyanobacteria. Phycocyanin is there. Phycoerythrin. C, phycoerythrin means cyanobacteria. Phycoerythrin. Actually, these xanthophyll, if we talk about it, somewhat slightly yellow in color, but chlorophyll A green in color. See phycocyanin. Phycocyanin is blue in color. Always remember it. Phycoerythrin is red in color. But we can call it as a blue-green algae because algae are photosynthetic, so we can call it as a algae also, and cyanobacteria is also undergo photosynthetic process. Why we use this term blue here? Because of the presence of more phycocyanin. Phycocyanin is blue in color and it is present in more amount. That is why we call cyanobacteria as a blue-green algae. Now, different oil droplets, proteins, these are scattered here. And this is the plasma membrane. Yes, here also gas vacuoles are there. And pigments are present all, and this is a thylakoid actually. Some cyanobacteria also have some special cells called heterocysts. And these cells are very specialized. Actually, nostoc, vena, all have the capability to fix atmospheric nitrogen. Oscillatoria, all are having heterocysts and they can fix the atmospheric nitrogen. Now children, one more thing, cyanobacteria, they are found in three forms. And so first is unicellular, second is colonial, and third is filamentous. Nostoc, vena, filamentous, colonial if we talk about microcystis, and prococcus, unicellular, filamentous, nostoc, vena, all are filamentous only. Now nutrition, what type of nutrition is present in cyanobacteria? So if we talk about the nutrition, we know that they are photosynthetic because photosynthetic pigments, especially chlorophyll A, which is the reaction center of photosynthesis. If chlorophyll A is absent in any of the plants also, they cannot carry out the process of photosynthesis. Remember it, because chlorophyll A only first absorb the particular wavelength of light to start this process of photosynthesis. So main thing is that they are having chlorophyll A, and along with that other pigments are also present. So they carry out the process of photosynthesis. That means they are autotrophic. Now reproduction. What type of reproduction is there in cyanobacteria? If you talk about reproduction, so vegetatively, they reproduce vegetatively as well as asexually. So that only we are going to discuss it. First, by binary fission. Second, we can say fragmentation. Binary fission, children, we know it whenever any, for example here, cell is there, single nucleoid is present in the center. Nucleoid means the chromosomes, or we can say DNA. Naked DNA is present here because in the bacteria's nuclear envelope is absent. So that is present in the center. Okay. Then later what happened in binary fission? It undergoes duplication. Duplication, that means replication of DNA takes place. Now it increases in amount because it replicates. It becomes double. Then later on, what happened in the next step? One septa appear here in the beginning in another diagram actually. So one septa appear here. As a result, what happened? It divide like this. Okay. Then later on, when it separates likewise, they completely, because of this common septa, they are completely separated from each other. As a result, two daughter cells are formed. Two daughter cells are formed. So this is binary fission, means breaking down of an organism into two. So by binary fission, they can reproduce by fragmentation also. Actually, fragmentation we studied in previous classes also, fragmentation in which an organism breaks into some pieces or fragments and each fragment then grows into new individuals. Similarly, cyanobacteria also. What happens? In that also septa appears. Septa means the demarcation. That in case of cyanobacteria, we can call it as a harmacodia. So these septas, for example, three septas are there. That means an organism breaks into three septa or three fragments, and each fragment grow into new individual. Then, after fragmentation, I am going to tell you about endospore formation, which is very, very important. Endospore formation. If we talk about endospore formation, what is actually endospore? Endospore is we can say the outer hard cover. Okay, as a result, the bacteria remain inactive for some time. So we can see in this diagram how endospore formation takes place. Endospores are of two endospore, or they may produce endogenously as well as exogenously, means within an organism or bacteria or outside the bacteria. Exogenously means outside, endogenously means within the bacteria. So here we are talking about the endogenous spore formation or endospore formation. Here replication of DNA takes place. These are the nucleoid, that is DNA, which get replicated. So these are in two forms. Okay, outer wall is cell wall, we know. Later on, what happened? Then it starts dividing. This is known as spore. And cytoplasmic membrane actually form spore. This is the spore. So this demarcation of septa appears. Later on, what happened? This vegetative DNA, this like vegetative nucleoid, we can say, it disintegrates. Disintegrates means now it breaks, but here it remains as such. Later on, it is covered by covering, which is made up of especially calcium and diphenylcholic acid and calcium. This structure we call it as a cortex. Later on, this cortex is surrounded by a pore known as endospore. And after that, as endospore form, then this outer lining that ruptures and these endospore are released out. So this is about endospore formation. And by the formation of outer cell, we can say outer hard covering also, they reproduce. This we call it as a akinase. Means by the formation of outer hard spore. This is known as akinase. Now I will explain mycoplasma. Bacteria that also comes under Eubacteria, heterotrophic bacteria. Heterotrophic bacteria means which feed or depend on others for their food. They cannot synthesize their food because they lack pigments. What? Photosynthetic pigments. Here, chemosynthetic means they oxidize nitrates, nitrites, ammonia. utilize for the formation of energy-rich compound ATP, and that is why they help in recycling of the nutrients. So heterotrophic bacteria, it is, some are useful also and some heterotrophic bacteria, they are harmful to us also. Okay, that means some are pathogenic. Cholera, that is also caused by TB, that is caused by bacteria only. TPT, that is also caused by bacteria. So these are generally pathogenic, and some are very useful also. Gut formation bacteria is used, that is lactobacillus. Even in the formation of we can say antibiotic also, bacteria are useful. They are useful for many more purposes also. Even we can say formation of beverages also, these bacteria are useful. Now I am going to tell you about mycoplasma. These are the smallest known organisms, living organisms. They lack cell wall. Cell wall is absent in mycoplasma. They are very small. Their size ranges from 0.1 to 0.5 micrometer. So they are very small. They even pass through the bacteria filters also. First of all, this was discovered by the Reuters. The name of the scientist is Ross in 1898. And where he found it? Actually, he found it in the cattle, those who were suffering from pneumonia, pleuropneumonia problems. Pneumonia which takes place is an infection in the lungs. That fluid gets filled in that lungs. So first of all, he only find it. Now, although they are not having cell wall, but they are having plasma membrane. The plasma membrane in mycoplasma is made up of lipids, proteins, and cholesterol. Here also in mycoplasma also that nucleoid is present, means DNA is naked, means without nuclear envelope. And if we talk about their nutrition, they are heterotrophic. They are heterotrophic in nutrition. They can survive in anaerobic condition. They survive without oxygen. They survive in anaerobic condition. These are pathogenic also. They can cause diseases in the human beings, different animals. So this all about mycoplasma, smallest known living organisms. They are also known as TPLO. You must learn the full form of TPLO, that is pneumonia-like organisms. Because first of all, the drops found it in the cattle suffering from pneumonia problem, in that fluid only. That is why it is named as pleuropneumonia-like organisms, very smallest known living organisms. Their size ranges from 0.1 to